Apart from that, Aditya. Okay, we will start our lecture. That is nothing but a chapter biotechnology. Okay, biotechnology, its processes and application. So this is regarding the last topic, that is applications of biotechnology. What are the applications? We already started the applications of biotechnology. For example, yesterday only we studied about the application of biotechnology in the field of medicine. Okay, means how insulin is synthesized. Okay, that are nothing but the first important application of biotechnology. Okay, means how that insulin is produced. So how that insulin is produced? The insulin, it is nothing but it is peptide hormones. Okay, that can responsible to maintain blood sugar level. Okay, so this is nothing but the insulin, and normally where this insulin is found within human body, it is nothing but the beta cells of Langerhans within the pancreatic cells. Now the insulin, it is made up of two chain, alpha and beta chain. Alpha chain, it is having the presence of amino acids. How many amino acids? Then it is nothing but it is 21 amino acids. And in the beta chain, there is a presence of 30 amino acids. Both alpha and beta chains get united to forms the insulin, complete insulin molecule. But when C linkage is bind with alpha and beta linkage, then that insulin molecule, it is termed as proinsulin, as it is present in the form of inactive form. But once proinsulin is converted into insulin, then that means insulin molecule is in the active form. So this is regarding the last lecture summary, that is nothing but the production of insulin. Okay, apart from that, in last lecture we already studied about what is vaccine, then what are the types of vaccine, that we are taking it orally, means oral suspension vaccine. Okay, the one concept is given in your textbook, that is nothing but oral suspension vaccine. The best example of oral suspension vaccine, it is nothing but it is it is nothing but the polio. Okay, so this is regarding our last lecture summary. In this lecture, we are going to study the applications of biotechnology in the process of gene therapy, okay, as well as in the process of formation of transgenic animals, in the process of formation of transgenic plant. And apart from that, one concept is given, that is nothing but the last one topic of this chapter. There are three terminologies are given only, that is bioethic, bioprivacy. So this is regarding our last lecture of the chapter biotechnology. So we are going to study what is gene therapy. But before gene therapy, there is one concept remaining regarding yesterday lecture, that is nothing but how these vaccines are formed. Okay, means जसं आपण last lecture ला बघितलं how artificial insulin is formed by using rDNA technology. That artificial insulin is called as humulin, that is the first insulin. So likewise, how vaccines are formed by using rDNA technology. So what is vaccine? Vaccine, it is nothing but the antigenic preparation of microorganism. So for example, we are taking hepatitis B vaccine virus. Okay, means hepatitis B virus, HBV, it is nothing but hepatitis B virus. So what happened? We are taking the genes from this virus. Okay, we are taking the genes from that virus which causes hepatitis. Now what happened? We are inserting this gene into the plasmid. Okay, so for example, this is PBR322. PBR 3T2. It is nothing but one of the plasmid. So what happened in the process of formation of vaccine by rDNA technology? We are taking the gene from this virus, that is hepatitis causing virus. Na? So we don't need all the genes. We are just taking only the fragment of gene. Okay, only the one fragment of gene we are taking, and this fragment of gene get incorporated into the vector. As this vector, it is already break by the enzyme restriction endonuclease. Okay, and here also the gene means it is nothing but the long chain, na? But we don't require this long chain, we just need this short fragment. Okay, so by the use of restriction endonuclease enzyme, we cut that DNA fragment. Okay, and as it is the virus, so virus means which genetic material is present in that. So as it is the virus, so the genetic material which is present in the virus, it is nothing but always RNA, not DNA. Okay, so we are taking this small fragment of virus, that is RNA, and it is inserted on this vector, and that is nothing but it is called as it is nothing but the process or steps of rDNA technology, that is the insertion of fragment from donor cell into the plasmid vector. See, I am denoting only P, that is nothing but the plasmid vector. So what happened after? After that, we are going to add this plasmid carrying the genes into the host cell. For example, this is host cell, and which host cell is mostly useful in the process of rDNA technology? It is nothing but the E. coli, or other than that, it is nothing but the yeast cell also used. But majority we are using only E. coli, that is Escherichia coli. So what happened? For example, this is E. coli. E. coli, it is having own genetic material. Apart from that, this is the recombinant DNA molecule, or this is recombinant molecule. Okay, now what happened in the production of vaccine? There are two steps are involved in, that is micro injection, okay, and fermentation. So these two processes are mainly carried out in the production of vaccine. See, this is not described in textbook, okay, but just for a general idea, I am just explaining that how these vaccines are formed within a laboratory by using rDNA technology. Now what happened after this host cell accept this plasmid, okay, that is recombinant plasmid or recombinant DNA or RNA molecule, this mixture then incorporated in the test tubes by using the process called as micro injection. Okay, the process it is called as micro injection. Now what actually happened in micro injection processes, the pipettes are taken, okay, and from that pipette, without sucking with the mouth, as it is the virus, okay, so from that pipette we are taking this mixture in a small test tube, okay, and we are keeping that test tube in a fermenter, and that is nothing but the process of fermentation. So what happened then? In the fermentation, the exact processes is going out and exact temperature is also supplied. See, fermentation आहे म्हणजेच काय? It is having the presence of fermenter, and the fermenter it is having certain suitable temperature as well as certain facilities, okay, to provide the conjugation. Means vaccine आहे. Vaccine means it is nothing but the antigenic preparation of microorganisms. So to avoid the outer surface, काय म्हणतात त्याला? To avoid that outer surface, what is the word, in the PTC, that is plant tissue culture technique also, we are providing the particular lab to avoid contamination. Okay, the word it is contamination. Okay, so in fermenter also there is specific suitable temperature as well as certain room is there, and within the lab the several facilities or certain availability is provided for the process of vaccine production after fermentation is happened. What that test tubes are after the process of micro injection and fermentation, that छोट्या छोट्या टेस्ट ट्यूब असतात खूप मायक्रो पिपेट सारख्या. ओके, सो विदिन दॅट टेस्ट ट्यूब a minute concentration of compound is present, and that is nothing but the vaccine. So in this way, for the vaccine production, we are taking the genetic material from the antigen itself. That's why we are saying vaccine. It is nothing but the antigenic preparation of microorganism. So in this way, vaccine is prepared in the laboratory. See, in exam, no one in your exam, no one is asking that what is the detailed mechanism of vaccine production. Okay, but for Neet concept, it is necessary to understand all. Now the next concept we are going to study about, it is gene therapy. Okay, one important point I am going to tell you that in your textbook there are so much application given. Okay, means subtitle is gene therapy, and in the previous lecture we started that the application of biotechnology in agriculture. So only one stanza is given in your textbook regarding the application of biotechnology in agriculture, but afterward in another pages there are several applications of agriculture again given out. Okay, so don't get confused regarding that. Just summarize in one paragraph and learn that. Okay, मीन्स तुमच्या बुक मध्ये खूप भारू दिलेलं आहे, तेवढं नाही वाचत बसायचं. जस्ट वन टायटल द्यायचं, अप्लिकेशन ऑफ बायोटेक्नॉलॉजी इन ऍग्रिकल्चर. ऍग्रिकल्चर मध्येच त्यात सगळं सांगायचं. इन द केस ऑफ ट्रान्सजेनिक एनिमल, इन द केस ऑफ ट्रान्सजेनिक. ओके, नाउ what happened in gene therapy? So gene therapy, the word itself indicate gene therapy. So gene therapy means what? It is one of the treatment. Therapy means it is treatment, but treatment about what? The treatment about genes. Now which genes? For example, there are certain diseases, okay, which are genetically diseases. So in that genetically, for example, which are genetical diseases? For example, color blindness, it is nothing but the genetical diseases. For example, emphysema, it is nothing but the example of genetical diseases. Other than that, diabetes, it is nothing but one of the example of genetical diseases. So all are nothing but the genetical diseases now. So what actually happened in the gene therapy? In gene therapy, we are just removing the defect gene, and on that portion we are adding the active genes. That is nothing but the concept of gene therapy. Okay, so what is the main purpose of gene therapy? Gene therapy, it is one of the treatment of biotechnology processes where the defected genes are removed out, and on that defected genes the new activated and recombinant genes are added. So that is nothing but the concept of gene therapy. So what happened in gene therapy? So see, in your book there are several points given out regarding gene therapy. Okay, so in shortly I am going to explain all the purposes, means why this gene therapy is used and what is their purposes. So the first purpose of gene therapy, it is nothing but in the clinical therapy. It is in the clinical therapy. Clinical therapy means, as I already told you, there are certain genetical disease. To treat that genetical disease, the defected genes are removed out, and on the portion of the defected gene, the new genes are incorporated. So that is nothing but it comes under clinical application. It is considered under clinical therapy or clinical applications of biotechnology. So what happened? एक second. The one of the example given in your textbook, it is nothing but SCID. Now what is the meaning of SCID? First of all, it is severe combined immunodeficiency. So it is nothing but one of the disease. Okay, SCID, it is nothing but it is severe combined immunodeficiency disease. So what happened in that diseases? So for example, see, severe combined immunodeficiency disease. So how this gene therapy help in this clinical format? For example, you know what is bone marrow? Okay, whether it is located bone marrow. Okay, so bone marrow, it is nothing but the part of the cells where our stem cells are present. Okay, stem cells are present in the bone marrow, and from that bone marrow there is a formation of our lymphocytes. From that bone marrow there is a formation of lymphocytes. Okay, means within a bone marrow the lymphocytes are formed. Now what is lymphocyte? Lymphocytes are nothing but it is the main primary organs of immune system. Lymphocytes are nothing but the main and important characteristics of immune system. Now this lymphocytes, it is again having two types, that is beta cells and T cells. T represent the thymus, B represented the bone marrow. Okay, that is B. So what happened? This T and B, these both cells are nothing but lymphocyte or lymphotic cells. Okay, now what is the function of this T and B cell? T, it is nothing but the cell mediated immunity, and B, it is nothing but the humoral immunity. See, there are several immunity which is possessed by immune system. Out of that, the cell mediated immunity, it is always carried out by T cell, and humoral immunity, it is always carried out by B cell. See, जर तुमचा human health and diseases, this chapter is completed, then and then you know what is lymphocyte, what is the type of lymphocyte, what is bone marrow. Okay, because in that chapter all are given, that is what is exact immune system, what is antigen, what is antibody, and all. Okay, so I am assuming that you all know what is lymphocyte, what is T and B cells. Okay, so what happened in certain cases is when the newborn babies are formed, these T and B cells are completely absent. Okay, so if these T and B cells are completely absent, then what happened? Then the baby is having no immunity at all. Okay, and no immunity means the baby is surviving from that diseases, and that disease, it is nothing but SCID na. And one more example, see, bone marrow synthesize this lymphocyte, both, that is T lymphocyte as well as P lymphocyte, but in the presence of enzyme ADA. This is one of the important enzyme. Okay, so what is the long form of this ADA? It is nothing but adenosine diaminase. Okay, this is one of the very important enzyme, and from or with the help of this enzyme only, this bone marrow secretes this T and B cell. Okay, but if this T and B cells are absent, so that person or that newborn babies are confirmly suffering from this disease, that is SCID. So what happened? By gene therapy treatment, or by using gene therapy treatment, the scientists are taken out the genes. The scientists are taken out the genes. Now this means what happened? See, the lymphocytes. Okay, the lymphocyte which are present on that पेशंट मीन्स पेशंट मधलाच lymphocytes काय करतात ते extract करतात, and on that lymphocyte there is a addition of this enzyme takes place, that is nothing but the ADA, and because of the insertion of this enzyme on that lymphocyte, the process of synthesis of T lymphocytes, B lymphocytes, and all can takes place automatically. So in this way, by using gene therapy, we can we can cure this disease, that is called as SCID, and this is nothing but one of the example regarding gene therapy. Okay, and this is important example regarding severe combined immunodeficiency syndrome. What happened? The lymphocytes already taken from a patient, and within that lymphocyte the manufactured, okay, the manufactured gene within the lab, that is adenosine diaminase enzyme is just incorporated on that patient's lymphocytes, and after inserting that, proper environment as well as proper treatment is going on, and after that the genetic mutation is occurred within that lymphocyte, and that's why the person is then capable of fighting all the diseases, as well as the person is getting the immunity. So this is regarding first example of gene therapy. Now the second example given, it is nothing but GH. The second example given in the gene therapy, that is nothing but growth hormone. That is nothing but the growth hormone. The second example which is given in your textbook, that is nothing but in gene therapy there is a production of certain growth hormones. Okay, for example dwarfism. Okay, dwarfism, it is nothing but one of the disease na. So in that disease the genes are modified. Okay, now genes are modified means there are several growth hormones. Okay, suppose GH, growth hormone. So these GH growth hormones is added into that gene, means patient जो असतो, within that patients these hormonal genes are added, and by the addition of that the dwarfism can be cured. This is nothing but the second example, that is growth hormone. एकदम डिटेल मध्ये जायची गरज नाही, because in detail these all portion are there, but for higher education, not regarding 12th standard. So just assume that gene therapy play an important role for the formation of growth hormone. Then the third point given regarding the application or purpose of gene therapy is to produce or to defect the somatostatin or tissue plasminogen activator. Apart from that, the main important concept regarding vaccine gene therapy, it is nothing but the production of vaccine, particularly which vaccine? Particularly the edible vaccine. Particularly the edible vaccine. Okay, so in this way gene therapy provides so many applications to cure the only genetical defects. Means what is the purpose of gene therapy then? Only to remove that defected portion of gene, and on that defected portion of gene just we are transplanting the new one, and that is nothing but the process of infusion. That is nothing but the process, it is called as infusion. So this is regarding the जीन थेरपी. सी तुमच्या बुक मध्ये खूप जास्त भारूड दिला आहे, तेवढं वाचायची गरज नाही. शॉर्टली तुम्हाला जस्ट आला तर 2 मार्कासाठी क्वेश्चन येईल, व्हॉट आर द ऍप्लिकेशन ऑफ बायोटेक्नॉलॉजी, देन यू जस्ट हॅव टू राईट इन मेडिसिन जस्ट एक्झाम्पल, then in agriculture, then again example, then again gene therapy, again in the process of transformation of transgenic plant as well as animal. Now the another applications of biotechnology is in the formation of transgenic animal. In the formation of transgenic animal, as we already study about transgenic plant. Okay, first we start the transgenic plant and then we are going to study about transgenic animal. Okay, so which transgenic plants are produced by the process of biotechnology? So for example, yesterday only I told you regarding BT. Okay, what is BT? It is nothing but Bacillus thuringiensis. And what are the example of transgenic plant which are formed by the process of biotechnology? It is nothing but BT cotton, then BT tomato, then BT soybean. Okay, then another also the examples are there, that is BT potato is also there. So these are certain examples of transgenic plants which are formed by the process of biotechnology. Now I'm just explaining only one example regarding all this. So for example, we are taking BC, means it is Bacillus thuringiensis. Okay, so what happened in this process? See, all these plants, and what are the main purpose of formation of transgenic plant is to act as a pesticides. Okay, is to act as a pesticide. Now what is pesticides then? Pesticides means to kill the pest, that is nothing but the pesticide. Likewise, insecticide is also there to kill the insect, that is insecticide. But in biotechnological processes, most of the pesticides are formed by the process of biotechnology, and one of the pesticides, it is nothing but the BT, that is Bacillus thuringiensis. So what is the mechanism of that? See, so BT, that is Bacillus thuringiensis, it is having the presence of BT gene. It is having the presence of BT gene, okay? And within that BT gene, there is a presence of cry gene or cry enzyme, okay? There is a presence of cry gene or cry enzyme, okay? And that cry gene, it is having the presence of protein. So what exactly happened in the recombinant DNA technology? So this is nothing but this is the normal process, that is Bacillus thuringiensis. It is one of the species of organism, okay, and from that we are just incorporating, or we are just extracting BT gene, and BT gene, it is having the presence of cry gene. Now what is this cry gene? It is nothing but it is the presence of crystalline protein. Cryogen म्हणजे काय आहे? It is nothing but it is the presence of crystalline protein, and this crystalline protein, it is favorable of alkaline condition. Means this crystalline protein, when gets alkaline condition, any alkaline condition. Alkaline condition means what? It require base phase, okay, not acidic, not neutral, basic, okay. So on that stage, what happened? This crystalline protein is converted into toxic material, okay. This crystalline protein is converted into toxic material, and that toxic material, when eaten by pest or insect or whatever it is, so automatically what happens? See, तसं directly होणार नाही, but for this process means this crystalline protein is get eaten by that pest. This cryogen, okay, this cryogen, first of all we are introduced in the plant, okay. For example, this is plant given. On that plant there is a incorporation of this BT gene is occurred. म्हणजे एक simple plant घेतला आहे आपण. Within that plant we are just inserting this BT cotton gene. After BT cotton gene, what happened? This plant, okay, this is nothing but the plant. So each part of the plant, it is having the presence of this cryogen, okay, because we are incorporating this BT gene onto the plant na. So what happened? If any pest or insect comes to eat this leaf portion or stem portion or whatever, generally for example caterpillar, अळीची पानावर येते, ती फक्त पान खायला येते. So best example given, it is caterpillar. Caterpillar, when comes to eat the leaves of this plant, what happened? The cryogen directly goes to the gut portion of caterpillar. Gut portion means it is nothing but the digestive portion of the pest or that insect, okay. So what happened? When that leaf containing cryogen is eaten by that caterpillar, then it goes to the gut portion of that caterpillar. Then what happened? The gut portion, it is having the alkaline nature, okay, it is having the alkaline nature. So automatically this gene gets favorable conditions. So in the gut, when alkaline nature comes in contact with that gene, what happens? This crystalline protein is converted into toxic material, and toxic materials are formed होत होते gut मध्ये. Then what happens? Automatically the death of that pest as well as insect occur. So in this way, by using rDNA technology, we can form various pesticides and insecticides. So this is the one example, that is Bacillus thuringiensis. Now the second example given, it is nematode. Nematode, it is also the one of the example of pest. Okay, so what happened in the nematode? It affects tobacco plant. It always affects the tobacco plant. So same process is carried out like that BT cotton. Same metabolism takes place. Okay, but only the difference is that this nematode, it is having the gene that carry the proteins. Means this nematode, it is having the presence of certain genes which synthesize certain proteins, and that proteins are important for the growth of the nematode. Now what happened in the preparation of transgenic plant? There is a preparation of cDNA. As I already told you, what is cDNA? It is complementary DNA, and it is made in the laboratory only by using gene library. Okay, so cDNA. So this cDNA play an important role in the life cycle of this nematode. Now, what happened? See, the general life cycle of nematode, it is nematode, it is having the presence of genes, certain genes. That genes are capable for producing protein, and that protein is important for the growth of this pest. Now, what happened for the formation of transgenic plant? We are getting, or we are incorporating this nematode containing gene into the plant. Okay, now after inserting this gene into the plant, what happened? The process of transcription and translation going on, and after the process of transcription and translation, this plant get replicate to form mRNA. Okay, now after mRNA, general process is there is a formation of protein, that is translation. Okay, but what happened by using rDNA technology? By using rDNA technology, we are adding this nematode containing gene here. So what happened then? The nematode containing gene forms cDNA, and this cDNA prepared in the laboratory. Now what happened? This cDNA and mRNA combine to form one conjugate, and there is no synthesis of protein is occurred. So no synthesis of protein occurred means no further growth of nematode is occurred. So in this way, we are making the pesticide. So this is nothing but the second example which is given. Then the another example which is given, that is nothing but hirudin. Hirudin, it is nothing but one of the anticoagulant. Okay, and this hirudin, it is found in one of the plant, and the name of the plant, it is nothing but Brassica napus, that is mustard. Okay, so whenever there is a formation hirudin, the name it is hirudin. Okay, hirudin, it is nothing but the anticoagulant. It is nothing but one of the anticoagulant. Anticoagulant means it helps to clot the blood. Anticoagulant. Okay, so hirudin, it is nothing but it is one of the anticoagulant, and it is found in the certain species of plant, that is nothing but the Brassica napus. So what actually happened in that process? The hirudin gene is collected in the laboratory. From that gene, there is a formation of seeds occur, and within that seeds, some oil is present, and from that oil, we are extracting that hirudin gene, and in a higher level of laboratory or at higher level research center, this hirudin is used as an anticoagulant for a particular time. No necessity to go the deep mechanism of this hirudin. Just remember the example. Now the another transgenic plant, for example, it is flavor saver tomato. Tomato, it is nothing but one of the best example of this transgenic plant. Now, how this is occurred? For example, see, if the tomato gets ready, okay, to bring into the market, what happened at that stage only? Tomato काय असतात खूप रेडिश झालेले असतात, रेड कलरचे, and means we can't preserve that tomato, means ripened tomato. The word it is ripening, okay, so we can't preserve that ripened tomato for a longer period. Means, for example, we are harvested from the plant that red color tomato, then after the means ते आणेपर्यंत बाजारातून ते घरापर्यंत जाईपर्यंत, so this is very lengthy process, and during that process what happened, this tomato is getted excessive ripening process, and because of that it get means ते खूप सडून जातात टोमॅटो. So to help out that problem of ripening, excessive ripening of that tomato, what happened in the process of biotechnology, by using rDNA technology, there is a synthesis of that several types of tomatoes are occurred, where we can preserve that tight as well as red color tomato for a longer period. Now, how this has happened? So see, it is nothing but tomato, it is having the presence of certain cell, na? So within that cell, the cell wall, it is having the presence of enzyme pectin, okay, and this pectin is always responsible to carry out the ripening process within the tomato. So scientists what did? So the scientists did this inactivation of pectin enzyme genes into that plant, okay, and the process is carried out by the using of again cDNA molecule, okay. So this is way the tomatoes are formed by using transgenic plant. Likewise, there are the example which is given in your textbook, that is nothing but the golden rice. Golden rice. So what is that golden rice? Golden rice means it is also one of the transgenic plant formed by the process of biotechnology. Now, what is golden rice then? Golden rice, it is the rice which is having high concentration of vitamin E. Sorry, vitamin A. Vitamin A ज्यामध्ये खूप जास्त असतात, that is nothing but the golden rice. And this rice, it is having somehow golden, means somehow yellowish look is there. And why this yellowish look is there? It is because of the genes which are present, okay, into that category. So this is regarding the formation of all the transgenic plant by the process of biotechnology. Okay, one point I just forget to tell you, that the name of this scientist, it is McGregor, okay, and that scientist formed that flavor saver tomatoes, means by inactivating the activity of that enzyme. So this is regarding transgenic plant. Now we will see what is and how many transgenic animals are formed by the process of biotechnology. So transgenic animal, first of all, what are the purpose of this transgenic animal? Okay, so the transgenic animal, the first purpose of the use of this transgenic animal, it is nothing but to study normal philosophy as well as development of organism. Means to study development and growth of organism, okay, means whatever the organisms are there, to study the development of the organism, we are creating this transgenic animals, okay. Then what are the second purpose of this use of transgenic animal? Is to study gene responsible for disease, means whatever the diseases cause to men. Okay, so the to study gene cause means whatever the diseases cause to the human beings. Okay, so to study that, we are taking this transgenic animals to study the genes of that disease causes gene. Then the next purpose, it is nothing but to obtain biological product. The next, it is nothing but to obtain the biological product. Now, which biological products are obtained by using transgenic animal? So, for example, sheep. Artificial sheep is modified by using rDNA technology. Now, what is again one of the cow, that is transgenic cow, means how many animals are modified or made by using transgenic rDNA technology? It is nothing but sheep, and the another, it is nothing but the cow. Okay, now see the name of the cow, that is called Rosie. The name of the cow, it is nothing but the Rosie. And the sheep. Okay, so what happened in the sheep? So see, why these sheep are used as a transgenic animal? Because the sheep, it is having the presence of gene, and that gene, it is nothing but the alpha 1 antitrypsin. It is nothing but the alpha 1 antitrypsin. So it is nothing but it is one of the, see, this sheep, it is having the presence of automatically this alpha 1 antitrypsin enzymes, and that enzyme, it is responsible to carry out several mechanism within, means several proper mechanism within human body. Also, the sheep, it is having the milk of this sheep. Okay, it is having the high concentration of the presence of this protein, that is alpha 1 antitrypsin. So that's why emphysema, it is nothing but one of the condition of the person in which the oxygen level goes down to that person's, means CO2 आणि O2 level fluctuate होतात, stable नसतात. So in that condition, the sheep milk is given to the patient. That is nothing but the one of the biological product formed by the use of biotechnology process by transgenic animal. Second, it is cow. Now, see, after the birth of offspring, some mothers can't able to feed that offspring, or certain issues are there. So in that case, this transgenic cow milk is given to that offspring or to that babies. Okay, because that cow milk, it is having the presence of enzyme, that is nothing but the alpha lactoalbulin, that is nothing but protein, one of the protein, alpha lactoalbumin. This is one of the protein which is present in this transgenic cow. So that's, and how this is present, this all process is carried out within the laboratory by using rDNA technology, and this process are first of all carried out in human, first of all carried out in animal, not human, and once they are checked, then it is provides to human. So this is regarding some of the biological products which are formed by the process of biotechnology by using transgenic animal. For example, certain vaccines are also formed. Just 5 minutes. Okay, for example, certain vaccines are also produced. Okay, but for the trial of that vaccine, directly not that vaccine, means that vaccines not directly giving to the humans. First, it is try on the animals. For example, it is try on the cow, it is tried on the rat, as well as on the dog also, because the genetic material or genetic combination present in dog as well as in cow as well as in the rat, it is somehow same, only one or two defects are there. So that's why whatever the vaccines are synthesized or produced by using rDNA technology is firstly trial on these animals only, and after the trial is completion, it is provided for the human being. And there are several drugs, for example in cancer therapy also, the high dose of cancer tablets are there. Okay, so first of all, that all chemicals are tried on the animals, and after the response are getting good from that animals, then it is applicable to the human being. So in this way we are completing applications of biotechnology in all the branches. So in this way we are almost completing the chapter. Just three terms are remaining, that is bioethnics and bioethnics, biopiracy and biomentalism. Okay, so in the next lecture when we are starting the new chapter, that is molecular basis of inheritance. So before that I just explained these three terms. Apart from that, we are completing all this topic of biotechnology. So hopefully सगळ्यांना समजला आहे का? Just give me response. Riya, Shraddha, Pranali, just tell me. आज जे सांगितले applications ते समजले का regarding biotechnology? Properly समजलेत का? Okay. Okay, so see, I think the time is over now. So in next lecture we are starting the new chapter, that is molecular basis of inheritance, and before that chapter just three terminologies are there which is remaining in this chapter, that we carry on. Okay, so till then, bye.